Good day, everyone. I hope that you are enjoying your Monday, March 9th, 2026. It's 11:01 a.m. I got a couple of announcements to make for you. The first announcement is your slide deck for today is up on our Canvas site. It's up as a PDF and a PowerPoint file. The next announcement is we will not be having any Zoom poll questions because I've fallen behind quite a bit on those. So I need to fix that. Um, the next thing is that Wednesday's meeting will be pre-recorded. So I will have a recording of Wednesday's lecture up before our class. So that means we will not be meeting during our regular Zoom time. So that gives you guys the opportunity to focus on other stuff for the remainder of the week. Or if you don't have anything, you can be like, you know what, I'm going to watch this. And then my spring break begins at that time. So that's all your call. I will post that before I'm gonna aim for posting the lecture. I'll aim for tomorrow night by 10 p.m. That's gonna be my deadline. But yeah, one of my friends who's an attorney, he told me great advice was promise low, deliver high. So he's always like, oh, put some deadline way out there and then beat it. But at any rate, um, before tomorrow night at 10 p.m., you will have your lecture for Wednesday. So we will not have a schedule or we will not have our regular Zoom meeting. I will have office hours and I will extend those office hours whenever you need them. Just email me and say, hey, can we meet at 11:30 on Wednesday? Sure. I'll email you back and say that works and we'll go from there. I'll email you back and say, hey, this is the link that you want to join. But with that in mind, today we're going to get after, and what we've talked about so far this semester, or not semester, but most recently, we've been talking about biochemistry. We've been talking about biological polymers. We've been talking about the building blocks of living systems and living organisms. And we haven't really gone in a backwards way, but um, we're introducing them all kind of like independently. Um, but they will all kind of blend together. So, most recently, what we talked about was sugars. We talked about sugar structures. Now, what we are going to do is we are going to talk about, let's see, yeah, good. We are going to talk about DNA. There we go. I start out by using or referencing DNA because I think that everyone has a little bit more context for DNA than the term nucleic acids. But if you have ever heard the term nucleic acids, consider yourself not fortunate necessarily, but you've got kind of a leg up there. DNA is an example of a nucleic acid. DNA stands for deoxyribonucleic acid. Now, all of these two terms, deoxyribonucleic acid and ribonucleic acid, these are common terms, common name terms. Um, deoxy or the D in deoxy stands for, sorry, the D in DNA stands for deoxy. So the difference between DNA and RNA is lacks and oxygen. That's the difference between DNA and RNA from like a name standpoint, and you could think about it almost like a functional standpoint. Now, I want to introduce to you guys a couple of things regarding DNA and RNA. The first item with respect to DNA or deoxyribonucleic acid, think about this as our um, carrier of genetic information. And RNA as kind of the interpreter slash translator. So there's DNA is like the long-term storage, whereas RNA is kind of a carrier that translates from that long-term storage to like the immediate needed information. Now, the first thing that I want to introduce you to that's kind of a historically significant event is an experiment that was conducted in 1952. This is known as the Hershey-Chase experiment. And the Hershey-Chase experiment, what you need to know, first of all, is this confirmed the theory that DNA, not protein, carried genetic information, carried genetic information.
Oh yeah, oh yeah, got the truth. Oh yeah, oh yeah.
So we have this thing known as a bacteriophage.
That's the truth.
My 5-year-old is sick today, but obviously not so sick that he can't play a video game and get really hyped about getting a trophy. Okay, so back to this. A bacteriophage. It's possible you've never heard this term before, but the way that I want you to think about a bacteriophage, and sometimes people use the term phage to replace bacteriophage. I'll say bacteriophage or phage. A bacteriophage is a virus that infects that infects bacteria. So another way to interpret this is you, a human being, cannot be hurt by a bacteriophage. But if you think about, your gut has bacteria in it. And if you get a bacteriophage or one of the bacteria in your gut are infected by a bacteriophage, you might that might impact you, but it's kind of like a secondary infection in the sense. So a bacteriophage is a virus that infects a bacteria. Now, this Hershey-Chase experiment was conducted to confirm, or well, it was really investigating how is genetic information carried from generation to generation. And in order to kind of pursue this, what the researchers who were doing this did was they said, okay, we know a bacteriophage, it's a virus, it's made up of protein plus DNA. And what we're going to do is we're going to put a little tag on proteins. So we're going to put an X. We're going to tag proteins. And what we are going to do is look at these bacteria and see if from generation to generation, does the tag go generation to generation? That's the question. So basically, here's this healthy cell. There's a healthy cell right here. And we're going to take some labeled or some tagged bacteriophage that have tagged proteins. And we're going to say, hey, bacteriophage, go infect those cells. So these cells right here, these bacterial cells, they get infected. That's what this right here is. So here is the infected cells. And the what the researchers did was they said, okay, well, what we want to do is we want to separate cells from the phage. And we are going to look and see, are the cells infected? Okay. Could everyone give me a Y in the chat if they understand that these bacterial cells are healthy, or an N if they don't understand that. Okay. So these are healthy cells. You give them these bacteriophage that have these little tags on them. And those tags are uniquely on protein. So these cells right here, are they H or I? H for healthy or I for infected? You got the arrows going on both sides. These right here, are they healthy or are they infected at this point in time? These guys have like this thing right here that I'm highlighting in yellow. That is what the bacteriophage has infected or injected into them. So these right here in the middle point, they are infected by the bacteriophage. Does that make sense? Give me a Y or an N if that makes sense. Okay. Okay. So these are infected. And here is where at the bottom, so they're infected. And so what that means is that the bacteriophage has injected whatever they need to inject into those cells to make them like to make the virus replicate. So at the bottom here, this is where it gets kind of technically complicated. We've got we're going to separate the bacteriophage from the infected cells. And we're going to look where is the protein tag. And the question really is beyond that, if the tag is with infected cells, what that means is that well, protein kind of brings the infectious agent. I'll say the infector. Infector? Yeah, we'll go with that. Okay. So that's the question here at the bottom. And the result of that was, well, cells, the bacterial cells, the infected bacterial cells don't have the tag. Does that make sense? Give me an X if that makes a little bit of sense, or any other character if you're mixed up on that. Okay. So essentially, this left-hand side of the screen is our way of saying that, hey, the bacterial cells are infected, but they don't carry any of that tag. So that means the protein didn't do anything to transmit that viral experience, I guess. Okay, so then the key is that this tag on the protein was uniquely on sulfur. And that's important because DNA has no sulfur. So if you're thinking, okay, what's the takeaway from this? The takeaway from this kind of or what's the takeaway as this stands for like how could I sum this up? And how is this done technically? I'll say magic sulfur was used. Sounds like a name of a street drug or something. Magic sulfur. Okay, so they used magic sulfur, and that magic sulfur didn't go with the cells. So if next Monday, when you're hearing people chant spring break, spring break, spring break, and someone's like, hey, tell me the coolest thing you learned in all of your classes, you can be like, well, I learned that magic sulfur was used in the Hershey Chase experiment in 1952. And what that proved was that protein did not carry genetic information. And that other person's going to be like, what? Magic sulfur? I didn't know magic was real. And you'll be like, yes, it is completely real. And it's called science. And they'll be like, that's crazy. You'll say, yeah, stop reading Harry Potter. That's fake magic. So let's move on with our lives. Okay, so now over here on the right-hand side, this was an investigation of DNA. Okay, is DNA the carrier of genetic information? And in order to do this, what they used is tagged or labeled phosphorus. So in this case, we can call it magic phosphorus. Now, I know that many of y'all are like fine arts majors and some business majors in there. I feel like whenever I say magic phosphorus and magic sulfur, what I'm really doing is I'm unleashing the untapped potential of chemistry and science's marketing capacity. Like, we need to start saying like magic phosphorus, magic sulfur, refer to everything as magic something, because then people are going to be like, huh? Magic? Fantastic. Okay, so we use this labeled phosphorus. You might want to call it magic phosphorus. And that was uniquely on DNA. So that phosphorus is not on protein. So protein, not tagged. Okay, so the exact same experiment was done simultaneously. You take bacteriophage that has magic sulfur, and then you take another batch of bacteriophage that has this magic phosphorus. You give them to each, or you give each of them to healthy bacteria separately to keep it separate so you can study a little bit more. Healthy bacteria, the healthy bacteria get infected. Then, okay, where is that label? Where is that tag? Where is that magic phosphorus or where is that magic sulfur? If it goes with the cells, that means that is the responsible thing. Magic phosphorus or this tagged phosphorus is what ends up with the cells. So ends with cells. It's almost like the people who are doing this experiment were like, okay, we're going to add one specific ingredient to check out this effect. We're going to use radio labeled or tagged sulfur. And is that, are these sick bacteria, do they have that sulfur? Yes or no? No, they don't. Okay, well, that means that protein did not contribute to carrying that genetic information. What about the magic phosphorus or the labeled phosphorus? Yes. Okay, there we go. That is what's responsible. But if you're looking at this and you're thinking, okay, well, what are the like two or three things that I need to take away from this? First of all, Hershey Chase experiment, 1952. Second of all, confirmed the theory that DNA and not protein carry genetic information. Okay, so that's our second detail. The third and fourth detail, you could just kind of consolidate it into one. And those are the identities of the atoms that are used, or identity of the substance that were used. Sulfur was used to label protein or to tag protein. Phosphorus was used to tag or label DNA. So that would be our third or our third and our fourth items. Okay, so Hershey Chase experiment. Is it DNA or is it protein that's carrying genetic information? And is it sulfur or phosphorus that carries that label? If it's sulfur, well, then that would say something about protein, but it's not sulfur. So phosphorus it is. Okay, the next thing, you probably have a little bit of familiarity with this, and this is the structure of a DNA molecule, also known as the DNA double helix. This was kind of finalized or theorized in 1953. There were three people who were recognized for and awarded the Nobel Prize. So what that meant is they took the cash money and they split it three ways. If I remember now, the cash award for the Nobel Prize is like $1 million. And so that's split between the recipients. So the recipients of the Nobel Prize were Francis Crick, James Watson, Maurice Wilkins. I believe all three of these, Maurice Wilkins might, no, I don't think he's still alive. But Jim Watson and Francis Crick, I think Francis Crick died within the past 5 years, and I think Jim Watson died in the past like 1 or 2 years. They share the Nobel Prize for that in medicine. The Nobel Prize is awarded for a number of different disciplines. It can be medicine, economics, um, chemistry, physiology. Actually, I think the medicine one is physiology. Anyway, they were awarded that. Now, this woman right here, I share her picture because what she did was she deciphered or took this picture right here. Now, this is what's known as an X-ray I'll just call it an X-ray. And this is an X-ray of you can kind of think about it as like a molecular X-ray. Um, her name is Rosalind Franklin, and she was a researcher studying, well, heritability, studying structures of DNA. She was a chemist, and she took this picture right here. And this is kind of recognized as being like, hey, without this picture, the theory on the structure of DNA would have remained pretty unknown. Would it have ultimately been resolved? Yeah, in time. But because of her work, these guys right here were able to kind of come to their conclusions. Um, and so it's kind of a, standing on the shoulders of giants that these three guys, I don't know if I want to say got just so it's not like they stumbled into it. Um, but they got this award in large part because of the work by Rosalind Franklin. Rosalind Franklin died, I think, in the mid 1950s. I think she died before the award was granted because, and the Nobel Prize isn't awarded posthumously. Um, but it does seem like, man, doesn't she kind of deserve a cut? And I think, if I remember correctly, the Nobel Prize is split no more than three ways. But at any rate, so these three guys were awarded the Nobel Prize for the structure of DNA. And there were other theories up until this point in time. Um, but ultimately, theirs was the one that kind of rose to the top. So 1953, DNA double helix. Rosalind Franklin is a big name because of her X-ray work. And then Francis Crick, Jim Watson, and Maurice Wilkins were the recipients. Now, this next thing that I want to bring up as kind of a very significant point in the history of DNA or in the history of genetics. This guy right here, his name is J. Craig Ventnor, Vent. I think it's Venter actually. Um, he is biologist, the geneticist, whatever you want to call him within that domain. He's a scientist, and he is one of the big figures in the Human Genome Project. Now, if you are unfamiliar, give me a one in the chat if you have ever heard the term genome. And actually, give me a one if you've heard genome but have no idea what the word means. And a three in the chat if, yeah, I've heard the word and I have a rough idea of what it means. Okay. So, this, whenever you see the word genome, what I think is helpful to do is to think about this as being two words: gene plus ome. Ohm would be kind of like global. So, or all encompassing. So the genome is kind of the collection of all DNA in an organism. And I say all DNA because there's some DNA that leads to genes and some DNA that doesn't. I always kind of think about the genome as being, if you think about like a map or something like that, if you look at a map of the United States, um, or if you drive on the interstate, say that you decide this weekend, I'm going to drive from San Marcos, where people live. I'm going to drive from San Marcos all the way to, let's go with, let's see, what's a far north? I'll just go with Iowa City. Why you drive to Iowa City? I'm not really sure. Maybe you really like farms. But if you were to drive from San Marcos to Iowa City, um, this is similar to the way that I think it's helpful to think about like DNA. There are parts of this drive along the way where there are cities. These cities are like genes. But then there's lots of space where there's nothing. I mean, there's a road, and there's definitely look to your right, look to your left. There's not really a whole lot there. Um, Much like DNA, it's like there are genes, and then there's lots of nothingness. Somebody sent me a direct message. I'm actually driving to Iowa City for spring break. Amazing. First of all, have a safe drive. Second of all, be cautious because I think that there's supposed to be a bunch of snow dumped on the Midwest in the next few days. But be very careful. And have a good time. Private message. Is it because they like farms? I'm going to venture to say maybe it's like family related. Um, but you do not have to share exactly why. But I'm really happy that I got to mention Iowa City and someone's driving to Iowa City. Um, Along the way, you will, if you haven't made this drive before, Oklahoma looks like Kansas. Kansas looks like Oklahoma. Um, I would get gas as far north in Texas as you possibly can. Just make it to Kansas. But that's just because, I wasn't born in Texas. I grew up in Texas. I don't like Oklahoma. That's one of the things that has been like deeply instilled to me, where I've had many people over the years tell me that I am not a real Texan. Okay, fine, that's fine. I respect that. Um, but I find common ground with them of like, I don't care for Oklahoma. And they're like, Joel, you got a chance. I'm thinking specifically of one of my friends from grad school. Okay, at any rate, now we're moving on. So my point is, in a DNA molecule, there's so much stuff. There are some parts of it that are genes, which is important, but then there's stuff that it's like, what separates those genes? And one of the things that's important to recognize is even though parts of DNA do not have genes, they're still equally important. It's kind of like, yeah, I want to drive from San Marcos to Austin. Well, what if, or San Marcos to Austin's not a great example. I want to drive from San Marcos to Dallas. Well, I need the road that drives through those parts of central Texas where there's really not much of anything. No disrespect to Belton, Waco. Waco is a city. But at any rate, I hope that you get my point here. So what this guy did and what this human genome project was there for was basically let's map everything. It's almost like there weren't any roads. And he was like, there's got to be roads. We got to find out how to drive through all of the genetic information that is in a human. And so this was completed in 2003. And this has been great because this is a tool that advances scientific research, advances cancer research, because now people can, sequence their genome through something like 23andMe and say, oh, well, I'm a carrier for um, this type of breast cancer. I'm a carrier. Okay, what are the chances that I'm going to develop this type of breast cancer? Well, they're better than this. Okay, what I'm gonna the way that I'm going to elect to kind of treat this in a preventative fashion is I'm going to be, you know, I'm going to get scans this frequently. I'm going to have an oncologist, even though I don't have cancer. And in the event that I develop this type of cancer, here's how I'm going to respond. So it's tremendously helpful and saves lives and prolongs lives. And um, so this work, it's not like, hey, let's draw a map of all the genes in the body and just for fun, because the funness I think wears out pretty quickly. There's value to it. Okay, so those are the three kind of foundational things. The Hershey Chase experiment tells you that DNA carries genetic information. The double helix, we got four people: Maurice Wilkins, Jim Watson, Francis Crick, and Rosalind Franklin. Then the Human Genome Project mapped everything out, and that's J. Craig Venter. So those are the three like foundational things that I want you guys to know about, because those are like, without those, where are we today? Um, Now let's talk about the structure of DNA. Well, DNA, like I said, is deoxyribonucleic acid. The difference between DNA and RNA is simply an oxygen. Now, that oxygen, we're going to start over here on the left-hand side. is within a what's known as a ribonucleotide. A ribonucleotide has this ring in the middle. This is a sugar. This sugar is known as ribose. Now, a ribo, hence that's where the ribo. Now, the next part is nucleo. Nucleo is going to come from this thing right here, which is simply identified as a base. We have what are known as nitrogenous bases. Now, if you've ever heard of DNA or had a class where you talked about DNA before, or RNA for that matter, are there specific letters that come to mind as like, oh yeah, something about the DNA code? If those do ring a bell to you, add them to the chat. If they don't worry about it. We'll get there. But these nitrogenous bases are what make DNA molecules and RNA molecules particularly unique. The last part, this tide, specifically the T there, is the presence of this phosphate. Okay, so a ribonucleotide consists of a sugar known as ribose, a nitrogenous base, and a phosphate. Now, a deoxyribonucleotide consists of a sugar. Okay, here's that sugar. Now I'm going to highlight the difference between a ribonucleotide, that right there, or that, and a deoxyribonucleotide. We said a moment ago the difference was an oxygen. Now, what I want you guys to know is a deoxyribonucleo, or I'll say deoxyribo. This sugar is known as deoxyribose. Okay, so that sugar is ribose, a nitrogenous base. And last but not least, a phosphate. Now, when you look at these two sugars, I want you to know that the difference between deoxyribonucleotides and ribonucleotides is not just an oxygen, but what's known as a 2' oxygen. So the presence or lack of a 2' OH. And I'm going to say 2' OH or 2' oxygen. That is going to be what helps distinguish these two different types of molecules. All right. Now, when you look at a DNA molecule or an RNA molecule for that matter, I want you to know a couple of things. First of all, I'd like you to, if I showed you this and this right here, if I showed you those structures, I would want you to be able to identify which one is DNA and which one is RNA. And you might look at that and think, well, I mean, I see DNA written to one, next to one, and I see RNA written to one. It's not for that reason. But instead, I want you to be able to look at these two molecules, the ones that I've got circled, and pinpoint that 2' carbon. And look at this and say, aha, that is my 2' carbon. And what does that 2' carbon? That is also a 2' carbon. And say, aha, well, this has an OH, therefore this is an RNA molecule. This one just has a hydrogen, therefore this is a DNA molecule. Could everyone give me a 5 in the chat if, yeah, I think I could do that. Okay. Now, when it comes to reading a protein sequence, we read protein sequences in their N to C directionality. When we read a DNA or RNA molecule, we read them in the 5' to 3' direction. Okay, so what does that mean? Well, if I were to list the sequence of this DNA molecule right here, I need to find the 5' end. That 5' end is where my 5' carbon is. And then I need to say, Well, here is my phosphate. Check. Here is my sugar, my deoxyribose in this case. But then this nitrogenous base is denoted by the letter A. So I'm going to label this sequence A. Then I'm going to go on to my next sugar. Here's my next sugar. Here's my next nitrogenous base, T. Then here's my next sugar. Here's my next nitrogenous base, G. Has anyone ever heard of DNA and heard about like ATG, CTG, ACTG, or anything like that? A genetic code or something like that? Or a DNA sequence? If you had, great, but if you haven't, don't stress about that. These single letters, the capital A, the capital T, capital G, those all denote different molecules. Those different molecules are your nitrogenous bases. A stands for adenine. T stands for thymine. C for cytosine. G for guanine. Now, if we look at this RNA molecule and we do the exact same thing, labeling the sequence in the correct direction, we're going to start at the 5' end and we're going to go to the 3' end. U, oh, well, U is uniquely found in RNA, not in DNA. Just as T, I'm going to put only DNA. U stands for uracil, and this is only an RNA. Okay. Now, this is kind of our sequence. So DNA, you read 5' to 3' for your sequence. Now, the type of link or the type of bond that connects one sugar right here to the next sugar is known as a phosphodiester linkage. That is what ultimately is going to give a DNA molecule. DNA has a overall negative charge. That's where that's coming from. Okay. Now, what I want you guys to know after that, got this slide in here, and this is just kind of I'll share this with you. Another way that you might see a DNA sequence written is indicating the presence of phosphates. So if you ever see something like PATG or ATGP, this lowercase p is indicating that there is a phosphate in the 5' position. This right here indicates we have a 3' phosphate. So I'll say has 5' phosphate. If you were to see ATG and nothing is written there, that would be no 5' or 3' phosphate. So what we see with this particular molecule, well, there's a 5' phosphate. So if we were going to write this sequence out as a DNA molecule, we would write it as P, capital A, capital T, capital C, capital G. These molecules that are in blue represent the nitrogenous bases, those nitrogen-rich bases that we saw a moment ago, simply denoted as the word base in a box or the single letter. Now, I'm not going to ask you to know the structures of these nitrogen-rich bases, but I want you to know that when you show, like, what is A? A is an example of a nitrogenous base. I've got the single letters and the full names for those nitrogenous bases right here. Okay, so the primary structure of a DNA molecule, you're going to list it in the 5' to 3' direction. You have phosphodiester linkages that connect the sugars. You have to be aware of the 5' end and the 3' end. Do they have a phosphate? Yes or no? Now, a DNA molecule will also have a higher order organization. This is known as its secondary structure. In this case, it's a little bit more similar to what you've maybe seen before. This is the DNA double helix. So this is that structure. And what is on the outside is the sugar phosphate backbone. So where you see this green, that's where these phosphodiester linkages are. So when you see the green ribbon, that's on the outside of the molecule. You have these two strands that kind of wrap around one another. And this green ribbon is going to be lots of negative charges. So I want you to think about a DNA double helix as having sugar phosphate backbone on the outside. On the inside or the interior of a molecule, you have nitrogenous bases, those A, T, G, C, and so on. And they're going to be hydrogen bonded to one another on the interior of the molecule. These two ribbons or these two strands will wrap around one another and they will make these major and minor grooves. So these are just kind of like contours and spaces that at a molecular level, something can get in here and read what is present on the interior of it. Thinking like a protein or something can get down there and be like, aha, there's the sequence. That's the sequence that I read. Okay. Now, let's go back for just a moment to this idea or this word nucleotide. A nucleotide consists of a nitrogenous base, a sugar, and at least one phosphate. I want to distinguish that from a nucleoside, which is only the sugar and the base. Now, nucleotides, you're going to see denoted in a couple of different ways. If the nitrogenous base is adenine, guanine, cytosine, or uracil, well, this is an RNA molecule. So what is your sugar in an RNA molecule? It's ribose. If you see adenine, guanine, cytosine, or thymine instead of uracil, you're talking about a DNA molecule. Now, your sugar there is deoxyribose. Deoxyribose. Okay. Now, when you look at these different, has anyone ever heard of ATP? Give me a 9 in the chat if you have ever heard of ATP. Okay. Possibly you've heard of ATP as related to energy or related to like, physical endurance or something like that. ATP is the energy currency of the cell. That's the molecule that makes basically your muscles contract because your body can use ATP for energy. Now, when we talk about ATP molecules, one thing that's worth noting is there's a difference between ATP and this lowercase d ATP. That lowercase d ATP is referencing deoxyribose. So DATP versus ATP, well, one of them is a DNA molecule, one of them is an RNA molecule. Okay. Now, what I want you guys to take away from this is I want you to be able to, if you saw GDP, I want you to understand that this G is referencing the base. The presence or absence of a lowercase d is denoting the sugar. Here you see there's nothing there. So that, with nothing in front of that G, that indicates you're talking about ribose. You're talking about an RNA molecule. Now, D and P. DP here stands for diphosphate. So that means two phosphates. The two phosphates that we're referencing are on the 5' carbon, and they are the alpha and the beta phosphates. If you saw GTP, well, that means we're talking about a triphosphate with a nitrogenous base known as guanine. And there's no lowercase d there, so this is an RNA molecule. So what I want you guys to be able to do is answer questions like, or sorry, answer questions where you take this and translate it. What sugar is in DGMP? And for that, I'd want you to be able to look at this and say, aha, lowercase d, that indicates deoxyribose. And then if I were to have a follow-up, well, how many phosphates does this have? I'd want you to look at this and say, aha, GMP. M stands for mono, so a single phosphate group there. Okay. Now, this is just kind of the nomenclature. Nomenclature when it comes to these numbers. 2' is referencing to the sugars compared to just the number 2. So if I ask you the difference between DNA and RNA, I said it is the presence or absence of an oxygen on carbon 2. That would not be correct because it's the presence or absence of an oxygen on the 2' carbon. Now, what maybe is the last part that I really want to stress to you guys, and actually the very last slide on here, various monophosphates, don't worry about that. This is just like, hey, there's different ways in which a nucleotide can be structured. This is in reference to numbers of hydrogen bonds. Okay. Here what we have is the nitrogenous base guanine and the nitrogenous base cytosine. There are 1, 2, 3 hydrogen bonds that hold together a cytosine and guanine, or cytosine and guanine molecules. So when we look here, and we have a G and a C, there's 1, 2, 3 hydrogen bonds. But then, if we look at this right here, adenine and thymine, 1, 2 hydrogen bonds. Now, this is important because, well, hydrogen bonds aren't pretty strong. But tell me in the chat, what's stronger? 3 hydrogen bonds or 2 hydrogen bonds? Add it to the chat. 3. 3 hydrogen bonds are collectively stronger than 2 hydrogen bonds. Now, this is important because if you think about, let's say you go on vacation or on spring break to Hot Springs, Arkansas. There's thermal vents there. There's water coming from the ground that is hundreds of degrees, no matter what time of the year. If you go to Iceland, there's thermal vents there. There's bacteria that live in those thermal vents. Could you live in an environment where it's 100 degrees Celsius or where it's 100 degrees, Celsius all year round? No, you couldn't. But these bacteria can. They can survive in those really extreme conditions. Why? How? Well, they're made up differently than we are. but also their genome. Their genome has a higher percentage or a higher, no, let me rephrase that. Has more GC than AT. And if you think about that and you say, okay, well, what's the significance of that? Well, let's imagine that you have a molecule that's 100, and the term for a DNA molecule is 100 base pairs. So that would be AT. So we got our two strands, AT, GC, 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 and so on. So this is a base pair. Tell me how many base pairs am I showing right here? Not hydrogen bonds, but how many bases pair up in that little box that I've got? Well, this is one base pair. Here's another base pair. Here's another base pair. Here's another base pair. So I've got four base pairs. Okay, could you give me a one in the chat if you follow that? Okay. Now let's imagine that I've got a double-stranded piece of DNA that is 100 base pairs. 100 base pairs. Um, and let's say that 70 of them, 70 of the bases are GC. So actually, let me revise that. Let's say that I've got one strand of DNA, and I'm going to tell you that 70 of these are Gs. So I got 70 Gs, G, 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 and so on. 70 G and 30 A. So I got A, and so on. So that's one strand. Could you give me a five in the chat if you follow that? Okay. So if I've got one strand that's made up of 70 Gs, 70 straight Gs, and then 30 As, what can you tell me about the other strand? How many G's does it have? How many C's, T's? How many A's does it have? And remember, these are opposite of one another. G pairs up with C's. How many G's are going to be in this strand right here? The opposite strand. 70 G's will, Elena, perfect. It's going to have zero G's. How many C's will this other strand have? 70, perfect. How many A's will it have? Zero. And how many T's? 30. Okay, perfect. So I want you to look at this and understand that one way I could communicate this is, or describe this is say, you have a 100 base pair strand of DNA. And I'll say it 100 base pair double helix. It is 70% GC. Could everyone confirm with a seven that they understand that, or a zero that they don't follow that? Okay. So we could say this is true about this molecule, 70% GC. Now, one question that I always like to ask related to this is, I'm going to clear all this out. No. 70% GC. I like to ask the question, what's stronger? which strand of DNA is stronger? A piece of DNA that's 70% GC or 70% AT? I'm going to go with A is our 70% GC, and then B is our 70% AT. And Elena, you're right. The one that's higher percentage GC is stronger. Now, fair question for that, or another way to think about that is, well, why? Well, if we've got 70 Gs and the opposite strand has 70 Cs, how many hydrogen bonds is that? It's 210 H bonds. And then we got to think about the T's. So there's 30A, 30T. Well, that's going to be 60 H bonds. So you can kind of think about it in this way. That's a total of 270 H bonds. Could everyone give me a Y or N if they understand this math right here? And I'm going to go ahead and 1, 2, 3, 1, 2. Okay. Whereas this one right here, well, 70 A's and T's. So I'm going to say 70 A's on one strand, and then I'm going to draw two lines to indicate two hydrogen bonds, 70 T's. Then 30 G's, 1, 2, 3, and 30 C's. So that would be 140, 140. And then that would be 90. So we're talking about a total of 230 H bonds. So it boils down to how many hydrogen bonds does each one of these does each one of these strands have? Could you give me a 9 in the chat if, oh, I think I could answer that sort of question, or a Z if I don't know how to answer that question. Okay. Now I'll take it one more step. And this last step is, I like to ask the question, which molecule is strongest? Molecule A, B, C, D. Molecule A is 10% G. Molecule B is 10% C. And let's just imagine they're all the exact same length. 10% G, 10% C, 10% A, or 50% G. Which one of these molecules is going to be the strongest? Okay, so let's look at molecule A. Again, let's do one strand and say 10% of it is G. That would mean if it's 10 base pairs long, it's going to be G. What about the other strand? The other strand's going to have a C complementing it. So you could argue that if it's 10% G, then it's 10% C. Does everyone follow that leap or that jump there? Give me a 5 in the chat if you follow that. If it's 10% G, it's 10% C. Okay. Now, 10% plus 10% would be 20%. So then the next part of this is that it's going to be, let's see, 10 plus 3, that would be 80% remains. So it's 20% GC, 80% A and T. So 40, 40. Okay. Does that make sense? So that gives us our ranking of this one. So it's, I can go ahead and stop right there with that. Now let's go on to option B. Well, if it's 10% C, just as we saw in the previous one, that means that it's 10% G, 40% A, and 40% T. So A and B are effectively the exact same. They have the exact same strength or the exact same kind of composition. So if A is right, then B is right. But let's go through the rest of these. Answer C. If it's 10% A, that means it's 10% T, which means it's 40% G, 40% C. Okay. So already C, by having a higher GC content, is going to eliminate A and B. Could everyone give me an 8 in the chat if they follow that? Okay. Last but not least is answer option D. Answer option D, 50% G. That means it's 50% C. So what does that mean? That means that it's 0% A, 0% T. So another way of putting this is it's 100% GC. So between these last two, D is the correct answer. Or between all four of them, D is the correct answer. Now, one thing that I want you to kind of understand with this is it's very tempting, but I don't want you to fall into this trap. Saying a DNA molecule is 50% G is not the same as saying a molecule is 50% GC. Could everyone give me a 3 in the chat if they understand that? Now, as a follow-up to that, if a molecule is 50% GC, what percent A is it? Perfect. It's 25% A. And therefore it's 25% T. Okay. Now, does anyone have any questions? Like this is the sort of question that I love to ask because it's a critical thinking question. You have to remember kind of which bases pair up with one another, and basically the number of bonds between them. But this is the sort of question that I want you to expect to be able to answer, or I expect you guys to be able to answer. That's what I got for today. Wednesday's lecture is going to be pre-recorded. I'll post that before tomorrow night. I've got office hours starting at 12:30. If I see an office hours, awesome. If you want to meet on Wednesday, we can do that. But like I said at the beginning, I hope you have a good break. Wednesday will be pre-recorded, so that will be up and we'll go from there. Other than that, though, thank you guys for coming.